So thanks everyone for being here. The DSF is my favorite organization in the world, and I'm just honored to be here, and I thank Veronica for asking me to speak today. So what I want to do today is give you an update on our work that we've been doing in collaboration with Stoke Therapeutics to develop this technique called TANGO, which is remarkable technique that stands for targeted augmentation of nuclear gene output, which you all know resulted in the drug STK-001 that is currently in clinical trials. So before I start, I want to acknowledge the group of just outstanding people in my laboratory who are very innovative and hard workers and dedicated to ending and solving Dravet and ending SUDEP. And this work, again, was done as a collaboration with our colleagues at Stoke Therapeutics and partially funded by a grant from Stoke Therapeutics to the University of Michigan. So we've been talking about this all day, and I don't have to introduce what Dravet is, except to say that this word cloud was made by a DSF mom a number of years ago to describe what their family goes through with Dravet, and to make the point that Dravet is more than seizures. It is a very complex presentation that needs to be considered holistically by physicians. And we are particularly interested in Dravet because of the increased risk of SUDEP of up to 20%, and the fact that in spite of novel therapeutics, that most patients with Dravet are intractable. And hopefully, with all of us here, with all these different kinds of therapeutic strategies, we can solve this issue. So as we have been talking today already, is that Dravet has a very, very complex presentation that is more than just severe seizures, but there is intellectual disability and developmental delay, movement issues, ataxia, growth defects, language and speech problems, mood disorders, chronic infections, sleep disorders, and so on and so forth. But the majority of small molecules really target the severe seizures in Dravet, but do not address these other complex and also important presentations. And so it became clear, it's clear to all of us in this room, that we need a better therapeutic strategy that targets the genetic cause of Dravet. And as you also know from these beautiful presentations earlier today, that variants in sodium channel alpha and beta subunit genes cause Dravet, and we know this. So here's a cartoon of what many years of work looking at structure and function of voltage-gated sodium channels tells us, is that sodium channels are heterotrimers. It means they're made of 3 different subunits. The central alpha subunit that comes together to form an ion-conducting pore, that's where the sodium flows. But then there are also smaller beta subunits that are also very important. And so everyone in this room knows that haploinsufficiency of SCN1A that encodes the alpha subunit called NAV1.1 causes Dravet, and that most of these variants happen de novo. But we also know, and I'm not going to talk about this today, but our lab has shown that inherited homozygous variants in SCN1B that encode this smaller subunit also cause Dravet through this loss of function, or it may be a separate and even more severe epileptic encephalopathy. Sodium channels are really important, and for neuronal function, we can't survive without sodium channels. In mammals, the upstroke of the action potential in a neuron is due to voltage-gated sodium channels opening, so that's critical. And sodium channels are expressed everywhere on neurons. And here's a cartoon of an excitatory neuron, and sodium channels are on the cell body and at the axon initial segment and nodes of Ranvier and myelinated axons, but also on the dendrites and on the terminals, on the synapses. And sodium channels are expressed not only in excitatory neurons, but inhibitory neurons, in particular fast spiking inhibitory GABAergic neurons that we know now are very important in Dravet syndrome. So this is an old slide by my colleagues, Jennifer Carney and Miriam Meisler from 2010, but I use it because you can actually see the channel. Now there are thousands of variants that are linked to Dravet and SCN1A, but here's a good example showing that variants in the N-terminus and variants in the C-terminus cause the same disease presentation. And early on, sodium channel biologists like myself were very intrigued by this. How could that happen? Until we understood that the majority of these variants cause loss of that allele or haploinsufficiency, and we learned also that 50% of SCN1A is insufficient for normal neuronal function. And that was also really surprising to sodium channel biologists, basic scientists who purified sodium channels, because we know in brain you have way more channels than you ever need. But for some reason, which we still don't quite understand, 50% of SCN1A causes this very severe disease. So actually, my postdoctoral mentor, Bill Catterall, made the first model of SCN1A haploinsufficiency in a mouse. He made it as a knockout in a mouse. And remember, back here in 2006, we didn't know, right, what the cause was. And this was the first demonstration that haploinsufficiency of SCN1A caused a disease. So these mice, here's the wild type, okay, and the nulls, complete nulls, and that's not the disease model, so they're just really severe. But this is a Kaplan-Meier curve showing that the heterozygotes, right, the haploinsufficient animals, underwent SUDEP. They had tonic-clonic seizures, and they had a loss of, or a haploinsufficiency of firing of the inhibitory neurons. And then when he looked at sodium current in the hippocampus, he saw that the excitatory neurons or the pyramidals didn't have any difference, but the bipolars, the GABAergic neurons, had a loss of sodium current in the HETs. You can see this little arrow right here. And he proposed at that time that decreased sodium current in bipolar neurons but no change in pyramidals caused disinhibition. And that hypothesis has lasted for over a decade now. But this is important because he did this in the mice at 2 weeks of age. Oh, remember this box? This is going to come up again later, this excitability curve of the inhibitory neurons. So these are the wild type here in the black, and then the heterozygous or the Dravet neurons are these guys that come up and then go down. So they have something called depolarization block. But Dravet is really complex. It's not only haploinsufficiency of hypoexcitability of inhibitory neurons, but there are other neurons that become involved with time. So when Bill Catterall did this at P14, he saw a selective loss of sodium current in the bipolar neurons. But when Jennifer Carney and Al George used a similar model and looked a week later, right about the time of disease onset, they saw increases in sodium current in the pyramidal or the excitatory neurons, and they saw that these neurons were spontaneously excitable. So there's a lot of different kinds of neurons that are involved in Dravet, and there are different kinds of ion channels that are changed in their expression and function as a result of that initial hit of haploinsufficiency of SCN1A. So we have to think, this is why it's so complex and such a wide-ranging disease presentation. So clearly we need a better therapy to get at this root cause of the disease, which is SCN1A haploinsufficiency. And this remarkable technique called TANGO, targeted augmentation of nuclear gene output, was the idea of Isabel Aznarez, who was a co-founder of Stoke Therapeutics. So she took advantage of something that was discovered a while back about many genes, especially those expressed in the brain, including ion channels. And these genes have something called a nonsense-mediated decay exon. It's also been called a poison exon by others, so if you read those papers. And so this is normally occurring. So if the exon is expressed, then this makes a non-productive mRNA that goes down the path of mRNA degradation and nonsense-mediated decay. So this basically goes down the garbage chute, okay? If it's not expressed and this splicing happens to splice this exon out, then it goes on and makes productive mRNA that goes on to make sodium channel protein, okay? And the brain uses this in all of our brains as a rheostat to determine how much channel is made. And it's also used during development. So early in brain development, you'll see this pathway happening, and then this one takes over, okay? So Dr. Aznarez had a wonderful idea, and that was to design an antisense oligonucleotide that sat on this NMD or poison exon and prevented this pathway and pushed the whole thing into the productive pathway, okay? And then into increasing productive mRNA and protein for SCN1A, okay? Makes sense. Let's unpack this a little bit more. What am I talking about? So SCN1A has these non-productive splicing events that are naturally occurring, and we can leverage them. So this NMD or poison exon, okay, exists. And so we know that if this exon is expressed, it makes a premature stop codon, okay, in the next exon and triggers the nonsense-mediated decay pathway. So this just goes to the garbage. But if we go the other way, then it goes to productive mRNA and protein, okay? So, and this event is conserved in humans, non-human primates, and mice, and we found it present in cell lines and brain samples from human patients. So this antisense oligonucleotide, or ASO, is designed to sit on the NMD exon, prevent this pathway, and push everything up into the productive pathway. Good on paper, does it work? Okay. So our colleagues at Stoke Therapeutics did a walk, an ASO walk along this NMD exon, which is basically called exon 20N, okay? And they made a series of antisense oligonucleotides of about 18 nucleotides in length and around every 5 nucleotides. And they tested them all. And they tested them for reduction of inclusion of the exon 20N, the poison or NMD exon, and increases in productive SCN1A mRNA, okay? And you can see this clear winner here by the arrows, which is number 22, okay? Which is the champion in preventing the inclusion of exon 20N, and it was the champion in increasing productive SCN1A mRNA. And the nice thing was we could either put it straight onto cells, and it didn't matter. You didn't have to have a special agent to get it inside cells. It would just go in, okay? And then we did a selectivity experiment to see if other sodium channel genes, which you can't read here, but these are the other main sodium channel genes, what this oligonucleotide did, and it only increased SCN1A with no effect on these other sodium channel genes. And the other really great thing was this sequence happened to be completely conserved in mice and humans. So it was immediately translatable. So this is an important concept, and that is that this ASO doesn't care what variant you have. It's agnostic to variant. So no matter what variant is causing haploinsufficiency, this ASO will upregulate the wild type or the control allele. So then we went into mice. It's nice to do things in cell culture, but we have to go into an animal. And so here we looked at wild type mice. So these are not Dravet mice. And we did a single injection ICV, so in one side of the brain, right into the ventricle, at postnatal day 2. Okay, that's the only treatment these mice ever got. And then in this experiment, we euthanized at 1 week later, and we looked at mRNA and protein. Okay, so we looked at dose-dependent loss of the non-productive transcript. You can see that. Dose-dependent increase in the productive transcript, and the important take-home message, dose-dependent increases in NAV 1.1 protein. So you can see that it happened. And then we did another very extensive selectivity, specificity experiment with all known sodium channel genes. And again, we saw effects only on SCN1A and none of the other sodium channel alpha subunit genes. Then we picked one concentration here, which was 10 micrograms from here. Okay, we picked this concentration. And then we did a time course experiment. So we gave, again, the animals only received one dose at postnatal day 2, and then we let them live to different time points, and then we measured the mRNA and protein for productive SCN1A and NAV1.1. And again, it was dose-dependent and long-lasting. All right, then we had to go into a Dravet model. And we chose Jennifer Carney's model because we knew it was very creatively made. So what Jennifer Carney did was she made a knockout of SCN1A on one gene background called 129, where there's very little phenotype. So these mice breed perfectly well and they don't seize and you can really handle them. But if you do an F1 cross, meaning we crossed it one time to another strain called C57 Black 6J, then we got the full Dravet phenotype. And if you'd like to look away while I'm showing these seizures, please do. Well, there we go. These mice begin to seize. Well, the earliest we've seen is the P14 to 15 range, but usually around the P20 range, you can see these spontaneous tonic-clonic seizures, which are very severe. And then the majority of mice undergo SUDEP. You can see this hind limb flexion here, and the mouse has died. So this is a very reliable model. Jennifer has shown a one-to-one correlation between this behavioral seizure and electrographic seizures shown on EEG. So there's spontaneous seizures, sudden death, or SUDEP, and cognitive and behavioral deficits. And you can see in the protein that there's about half of NAV1.1 sodium channels. So here's the important take-home message that Chungling Chen in my lab did. So these are in the DS mice that I just showed you. And she took cohorts of these mice and injected them in postnatal day 2 one time. And we decided to go with 20 micrograms ICV of the ASO to make sure that we would have enough coverage. So she injected at postnatal day 2 in one side of the brain. We began monitoring them by infrared video camera 24/7 at postnatal day 17. And then we euthanized a cohort at 7 weeks and another cohort at 14 weeks to measure mRNA and protein. But you can see the remarkable result here. And that is, so we injected mice with either vehicle or the ASO, and the vehicle was just a PBS or saline. Here's what you've seen now a couple times from my colleagues talking, and this model has a remarkable rate of SUDEP. So at around postnatal between 20 and 30, we lose about 80% of these mice to SUDEP, just like I showed you in the video. The wild-type mice are here. Nothing happens to them. And the good thing is, if you inject ASO into the wild-type mice, nothing happens to them. They're fine. And what was incredible to us, that in this experiment, we lost one DS mouse, after our treatment with the ASO. So the ASO changed these mice from 80% SUDEP to maybe about 1%, okay? And then when we looked at 7 weeks and 14 weeks, we saw increases in mRNA and protein, okay, of SCN1A mRNA and NAV1.1 protein. One thing we did see was that the treated wild-type mice also had increases in mRNA and protein, and actually had increases above control in their levels of channel. But these channels must not get to the plasma membrane and are probably not functional because we didn't have any toxicity in those wild-type animals. So even though you have too much NAV1.1, it doesn't seem to be detrimental in this model. Okay. And then we instrumented a cohort with the same experiment. So we injected at P2 one time, and then we did surgery to put EEG electrodes, you can see the cartoon here, at postnatal day 20 or so, and then we monitored them. And we looked at the numbers of seizures and the latency to first onset. So you can see here very clearly versus the control vehicle injection that there's a significant reduction in the number of seizures in these mice. There's also significant prolongation of latency to first seizure in these mice. And here's some examples of the EEGs. So here's a wild type, right, with PBS injected, so nothing happens, right? And here is a DS mouse injected with PBS, and you can see this electrographic seizure. Here is an example of a DS mouse after injection with the ASO. We did not see evidence of seizures. Rarely, however, we did see evidence of seizures in the DS mice that were treated with the ASO, and when we did, these seizures were not distinguishable from the untreated mice. So there is a breakthrough in seizures that can happen, although it's very rare. Okay, so recently we wanted to know, how does this work, right? What is the physiological readout in neurons of the action of STK-001? You've heard about this from Stoke Therapeutics. That's the drug. We are now using a surrogate that is a slightly different ASO that has the same biological function in mice, because this one, as you know, is in clinical trial. So we crossed our mice with another mouse that had a PV TD tomato reporter. So what that means is these mice glow red, so that they're fast-spiking interneurons that are parvalbumin positive glow red under the microscope, so that the investigator can see them while he or she is recording from the brain slice. You can see an example of how we do electrophysiological recording. So we're doing this in real time. Now remember I told you when Bill Catterall first looked at his mouse, he looked in the hippocampus and he saw hypoexcitability of the GABAergic interneurons, but he was not able at that time to tell us whether they were parvalbumin fast-spiking interneurons or not, because that technology wasn't available. So we're looking directly at the PV neurons, but we're looking in the cortex now because that has a greater correlate to Dravet syndrome seizures. So you can see here, as we expected, so here, these are action potentials that are firing at different current injections, and this is a wild-type mouse. And here's a Dravet mouse, so this is a brain slice. And one thing we noticed was that they fire at lower current injections. So my gosh, they have a lower threshold for action potential firing, but then they also undergo something called depolarization block. So they get blocked here at higher rates, or higher current injections. And here's another example of depolarization block in the Dravet neurons versus the wild type. So you never see that in the wild type, and you reliably see it in the Dravet neurons. And if you quantify that, you can see it here. And this is something that no one has ever reported and is consistently, significantly, statistically significant in our hands. And that is, so here's it's called an input-output curve. So we inject current and we count the number of action potentials in the PV neurons, and that's a wild type. And this is in the Dravet. So there's an initial hyperexcitability of the fast-spiking interneurons. And then with higher current injections, they undergo depolarization block. And that was similar to what I put in the red box from Bill Catterall's work in the hippocampus. Okay, now, these mice were treated at P2 with a single dose of STK-001 surrogate, okay, in one side of the brain, ICV, and then we recorded them around P22 to 25, and that's when you see those big seizures. That's the time of disease onset. And one thing we noticed was they were firing more normally, and they certainly no longer underwent depolarization block. And when we quantified the results of a lot of experiments, we saw this. And this side is, these are wild-type mice, and this is important as well because the wild-type untreated are here in black, but the wild-type with the ASO, those are in blue. So there's really no difference. You add this ASO and it does increase the number of channels in the wild-type mice, but it seems to have no effect on the overall firing pattern. And here, with the DS mice, you see that the DS untreated or the PBS treated have that hyperexcitability followed by depolarization block, but the ASO treatment normalizes that and takes it back to the wild-type firing pattern. We've also been separating our mice by sex, male and female, and it appears that the STK-001 surrogate affects males and females equally. And then we looked at the action potential properties. So here's a typical neuronal action potential. So this threshold, this upstroke here is due to the opening of sodium channels, and the repolarization or the downstroke here is a combination of sodium and potassium channels. And we measured that. So in the untreated DS mice, we saw that the action potential was wider. So this width of the action potential is wider in DS animals compared to control. And the maximum rate of rise, or how fast this rises, is slower. And the maximal rate of decay, or how fast it repolarizes, is slower. So that implies that there are sodium channel differences, but there are probably potassium channel differences too as a result of that haploinsufficiency. But the nice thing is these are all non-specific, these are now indistinguishable from each other. So in the STK-001 surrogate treated animals, all of these action potential properties normalize. Finally, we want to know so the output of inhibitory neurons is to release GABA. So what happens in the DS animals versus the surrogate treated animals? So here we looked at, these are inhibitory postsynaptic currents. Okay, so we are measuring what happens in the postsynaptic pyramidal cell, or the excitable cell, as a result of the influence of the inhibitory neuron, right, which releases GABA right here, okay? And downward deflection is an inhibitory current. So in the wild type, you see all these really nice inhibitory postsynaptic currents, but in the Dravet mice, and here, this is in the cortex, you barely see any. So the GABAergic tone is really pretty much gone in these animals, and you can see it quantified here in that the frequency is severely different, okay, in the DS versus the control, but the amplitude is not, and that means to an electrophysiologist that it's release from the GABAergic terminals onto the pyramidal neuron that is impaired. But here are the STK-001 surrogate treated animals. So you can see the wild type, okay, lots of inhibitory postsynaptic currents, and then the DS animals treated, lots of inhibitory postsynaptic currents, and in fact, they are not distinguishable from each other. So this is restored to this by STK-001 surrogate and appears to normalize GABAergic signaling. So our new work shows that a single ICV treatment of STK-001 surrogate at postnatal day 2 restores excitability and action potential firing properties of the cortical fast-spiking interneurons in Dravet mice. Those are the PV neurons. It affects female and male animals similarly, and it restores GABAergic signaling in the DS cortex. And so when you think about, kind of a simple way to think about epilepsy is disruption in excitatory inhibitory balance. You can think about this teeter-totter. And in Dravet, the excitatory tone goes way up because the inhibitory tone is lost or disabled. And what we have shown in our work is that this inhibitory tone is restored. You've all heard about this from our colleagues at Stoke Therapeutics, that STK-001 is in a number of ongoing clinical trials. And their initial data that were released at the American Epilepsy Society meeting shows a reduction in seizure frequency, especially in the 2 to 12-year-old cohort, although this is ongoing and we have lots more work to do. So in conclusion, Dravet is more than seizures. You all know this. And a holistic evaluation of DS patients is really important to treat the complete disease presentation. And transgenic mouse models, and all of these models are invaluable to understanding disease mechanisms across multiple organ systems. And this basic science provides the preclinical foundation for the development of novel therapeutics. So the TANGO approach may provide important therapeutic benefit to a wide range of DS patients with SCN1A variants that result in haploinsufficiency, which means truncation, nonsense, frame shift, partial or whole gene deletions. But for clinical trials, the patient must be evaluated for their variant. Because as we just heard this morning, gain-of-function variants should never be treated with this ASO because they would increase the expression of the gain-of-function variant and probably make the epilepsy worse. So this work has resulted in clinical trials that are ongoing. So with that, I'd like to thank the remarkable group of people in my lab who have done this work and our wonderful colleagues at Stoke Therapeutics, also our funders especially, NINDS. Thank you.
All right, we're going to take a few questions. I'm going to hand it off.
Lori, again, as always, amazing work. Very exciting. So I just have a couple of questions.
Okay.
The data that you showed, you injected the ASO at P2. Of course, I think it's starting from P14 is when you see the curve starting to get nasty in these mice, which is kind of with humans. P2 is really early.
Really early.
I'm wondering if you've tried to inject it at P10, P14, and see if you can rescue it then too?
We did. So in the Science Translational Medicine paper, we did that experiment over again at P14. And we did get a significant reduction in the suit-up rate, but it wasn't as profound as the P2. We did try to push it further, and it just kind of goes away. But the problem in this model is you have such a little window, right, to put in therapeutics.
Yeah, and then my second question is, so you especially looked at the PV, the effects on the PV cells.
Yes.
And that data is cool, but I don't think you mentioned, are there any effects on the somatostatin cells as well?
So we are doing that. So what we have to do, we have to finish crossing to the somatostatin marker. So we're crossing to that, and we're crossing to VIP, so we can do that. So as you know, it takes months to do it. But we're also doing a whole series of experiments on the excitatory neurons because NAV1.1 is expressed there too. So next year. Thank you.
I think we can still do one more question if we have one. Anybody?
Thank you for your time. Did you guys notice that there was any decrease in the comorbidities besides seizures with this treatment?
Yeah, we're doing that now. We have noticed some differences in the ataxia. We're also doing all of the behavioral experiments now. It just takes a long time to do it. So it's coming. Thank you.
Great, thank you.
All right, thank you.